Okay, so welcome for this last lecture for today. Actually, I prepared something a bit light because I know it's the end of the day, middle of the course. We are all tired, so I will try to make something easy for you, and I hope enjoyable. And this is why I'm not going to talk that much of my work. I'm going to talk mainly of the work of the other, and maybe in the last 10 minutes, I will tell you what we did. But actually, the model is quite simple. So I think in 10 minutes, you can then get the idea and understand what is going on. So it's better to have a kind of a background about what I'm going to present. So what I want to discuss, it's a bit in the continuity of what was presented in this morning about dynamical system, but now with the addition of space. Because if you take biological system, actually, when you look at them, you see that they are highly organized in space. And one of the concepts is this idea of polarity, where actually this polarity means that you have an asymmetric distributions of molecules, activities, compounds in cells or in organisms. It's at every scale, from a single cell to organism. Here I draw a couple of examples that you find. This is actually the asymmetric cell division that we discussed. We had a question in the last presentation before, where one of the daughter cells is a neuroblast, is going to be a stem cell, while the other one is differentiated. It's a way to differentiate. And one of the compounds is localized in only one of the cells. Here you have the Drosophila embryo that has this morphogen gradient that will pattern the organism along its AP and DV axis. You have an example of a single cell, a migrating cell. I'm going to talk a lot about it, where you have a front and a back of the cell with the axis of polarity aligned with the migration axis. This is an example in tissue, again, the planar cell polarity, where every cell is aligned according to one axis here. This is more to show you this apico-basal polarity from the, between the lumen and the apical, the basal and the apical surfaces of cells. And this is again in a single cell in an organism where you have this par 3, par 6 asymmetry in the C. elegans embryo here in that case. So this is just a picture to show you that all these biological systems are very well organized in space. And when you think about it, it's not easy. I mean, if you take an acid tube or a piece of water and you try to mix stuff inside, you shake it. And at the end, you get something mostly homogeneous. So it's pretty hard to make something that will actually show pattern. So that's a big question for in the term of cell polarity. So what are the physical mechanisms behind this emergence of pattern? And before I get a bit further, actually, as a physicist, and I show you two objects. So I will do like Leila, make a little game. So I have two objects, one magnet and one pencil. And I just want you to, I will ask one question. So for whom these two objects are polarized? Can you raise your hand, please? Yeah, maybe a little half. So for whom only the magnet is polarized? One. Actually, as a physicist, I would say that only the magnet is polarized. You know, this pencil is not a polarized object. This is what actually Lewis-Wolper claimed here in this paper, because the polarity means something that has to be deeply, let's say, written in the material itself. You know, if you take a magnet, you break it in two parts, then you have two magnets. So it's something where really every piece of the material has this polarity axis embedded in its microscopic part. Whereas if you take a pencil, you break it in two, and you don't get two pencils. You know, you have like one side that will still have that, the other one which will be able to be erased, but you don't get two pencils here. And the idea is that this will be shaped by some elements that will make it asymmetric at the end, but that you cannot call it polarity. And I thought a lot about these two objects. And I think at the end, in biology, you have a whole mixture of polarity that goes from this magnet to the pencil here. Just to give you the two most extreme cases for me, the hydra, for example, is an animal, you cut it in two, and then it will reassemble itself, making a new head and a new tail. So it's a perfect magnet somehow. Every cell here knows where it is, where it's sitting, and whether or not it has to become a tail or a head. But on the other sense, if you take embryogenesis and this early experiment from Spearman and Mangold, where you can take a piece of, like at the very early stage here, you chop a piece of tissue, and then you glue it under embryo, and you end up with, in that case, a fish which has two heads. So it's really showing you that you have this kind of inducing centers so some pieces of the material that are different from the other one, that is sufficient to make the whole head away from it. So it's really, you have the way to shape this object here and wherever you want. You can make it on the bottom here as well. So I do think that polarity is really in between these two extreme cases here. So if I go back to this first picture of all different polarity in biology, I'm going to talk mainly today actually about cell polarity. So the polarity for just single cell. And where the main question is going to be, I have an object, which is like a couple of microns width, and I just want to make something asymmetric within it, to have a patch somewhere here. And just to give you some example why this could be interesting. So if, for example, if you want to do division, so you want, for example, this is a yeast, and you want to make a bud somewhere, so you want to divide in two, and to segregate your genetic material in two pieces, you want to have one of these patches somewhere here. If you're a migrating cell, the problem is the same. Imagine that you want to have a front that is going to be the leading axis, and that will basically track the cell toward this direction. Again, it's the same principle. You want to have one patch somewhere. So that's kind of the whole idea of this course is which kind of model, which kind of physical mechanism can do that here. So if you just look at biological systems here and you just kind of ask what are the biological requirements for that, just very easy principle. The first thing that is important is to have a unique, in this case, a unique polarity axis. So of course, if you want, for example, to divide just in two, you don't want to divide in 4 or in 3 because you cannot divide your genetic material in 3 or 4 cells. You just want to have one patch. You need to have one axis of polarity. In migration, you want to move forward. If you start to move if you have a cell that has distinct activity patches here, then it cannot really move coherently. Okay, you want that to resolve in a single front that will give you this unique polarity axis here. So the second requirement that you could ask for is actually to have some, this is like uniqueness, let's say, to have something that will show plasticity. So what I mean by plasticity is that actually most of these cues that we will localize somewhere, they are maintained as a steady state. They can actually be reoriented as well. So this is particularly important during migration. So you also, I guess, this movie of a neutrophile that is chasing a bacteria. So in a sense, the cell there's a new signal that is coming here. The cell has to reorient its polarity axis and then start to move in this new direction here. So this plasticity is, let's say, is the sensitivity to incoming signals. So the third thing will be, and it's a bit in balance with this idea of plasticity, is this idea of robustness. So robustness, because actually you want the cell to be, and this is something we discussed a lot this morning as well, to not be sensitive to filter out fluctuating signals. So you don't want to have like a little perturbation that will completely screw up your pattern. Okay, so you want something that will be able to filter out and to don't amplify the little fluctuations. That could be like fluctuation of the environment, but the robustness can be also thought about the robustness against the internal variation also. It has to be robust against variation in concentration, for example, of concentrations. So concentration, for example, if you double the amount of a protein or you change it by a factor of 10%, you don't want that also you are going to lose this property here. And here you can say also this robustness should be about every kind of details that doesn't count. For example, the detailed shape of the cell, if it's not important, any kind of details like that. Another important fact, I'm almost done about this exhaustive list, let's say, is the scaling issue, which is particularly important. The scaling is that, you know, if you have a small cell, you want to have one unique site like that, and if you have a big cell, you also want to still have one site here. It looks a bit trivial, but when you think about physical model, this is not easy at all to achieve, this scaling issue, and this is really an issue here. And the last point here will be also the ability to make spontaneous polarity. So this is not always the case. Sometimes this polarity is really driven from the outside, but most of the systems that polarize, they can do that by themselves. Okay. So they have the ability to break symmetry, to do symmetry breaking, and to self-organize and show this emergence of polarity by its own, just the internal consistent enough to make it appear. So this is a bit listing, because I just wrote it on the blackboard, because I will come back presenting the different models where they fail, actually, on those different aspects here. So this is a bit kind of the first point from biological aspect. The second point will be more from physics, actually. I'm going to show you what are the basic way that physicists thought about how to pattern an object in space. So I'm going to talk about patterning. And patterning is really an idea that is really related to cell polarity, because at the end, what you want to do is to pattern space when you polarize an object, a biological object here. And for that, I will start with I think, what is the most important idea behind that, which is actually related to self-organization. And let me just start with this very simple videos of chemical reactions that I like, that for me was one of the most important things to like science a couple of years ago, like more than 10 years ago, actually. And this is the Belousov-Zhabotinsky chemical reactions. I don't know if you know these reactions. So it's a chemical reaction, which means that here you have a mixture. So this is like a plate that you see, which is about, it's a Petri dish of about 10 centimeters, where you have 5 chemicals that are going to be out of equilibrium, which means that you inject actually in the system everywhere, like in a diffuse manner, one of the reactant all the time. So that will be consumed by the reactions. And actually, if you look at these chemical reactions, you see this beautiful emergence of spatiotemporal order. So you see this kind of spiral that forms. And when you think that this is a chemical reaction, you know, it's something where actually the elements that are playing the game here are molecules, okay? Like the molecules are much, much smaller than what you see here. This is 1 centimeter. So how these things emerge that are microscopic from microscopic rules, this is, for me, was a big mystery. Now we understand very well how it works here, and I will try to explain a bit what's behind that. But for me, this is the idea of patterning, and what I discussed a bit before, how you can make patterns that emerge from just like a bath of chemicals. Yeah. So I just let it go. So if you want to understand that, we are going to discuss about actually reaction diffusion equations. And just to make a link from what we saw before, to put myself in, so this is a diagram that I like, where you have in this axis the number of variables, so 1, 2, 3, up to the continuum. And here, from going from the linear to the non-linearities in the system here. And actually, so you might see some of the systems that you know very well. The exponential decay or growth here is a linear system with one variable, very simple one. You can have more variables to get oscillations, up to like a continuum where you can get waves here. And for the non-linearities, we had many examples this morning again, with instabilities, limit cycles that you find here. And actually, the reaction diffusion I'm going to talk about are really like in this scheme of like a continuum and a non-linear. So this is what people call in general a spatiotemporal complexity, in a sense. And this is why it's a bit hard to tackle with mathematics, but I will try to explain it with hands, mostly, today here. So the first model that I will comment, and maybe some of you knows, but for the one I don't know, it's a very important one, I would call them the Turing-like patterning system. And that was actually founded by Turing, who was the first one to propose how you can make this pattern emerge, spatial pattern that emerge. And his idea was pretty simple, is to take a scheme where you have two molecules, a molecule A that has autocatalytic activity on itself, so something that will activate itself. And also this molecule A is going to produce a molecule B, which should be an inhibitor, actually. And this inhibitor has a retroaction on A and inhibits A itself. And the whole idea of Turing was to say that actually this production from A to B is something local, and the inhibition of B to A is something that is global. Yeah. And you will see why this is important. Yeah. And how you make that happen, actually. And the whole idea is that if you think about, like a acid tube, the idea is to suppose that these molecules A and B has two diffusion coefficients, which are different, such that the diffusion coefficient of particles B is much greater than diffusion coefficient of particle A. And with just that rules here, actually, you can get this emergence of patterns in space. So you can, there is a mathematical way, I will explain it to you in a second. I will just explain it to you just on the drawing, how that works, actually, on the blackboard. So the idea is pretty simple. So let's assume that this is an axis. Well, you have this is space on that axis, and this is for concentration. And imagine that you start with some concentration of A's, uniform but a bit variable, because the uniformity doesn't exist. There's always some fluctuation. Like if you go to the molecular scale, you still have some Gaussian fluctuation just due to the finite number of particles. And then you also have a concentration of B molecules, like that here, at the beginning. And the idea is that now you consider this, local production of A and self-activation of A. So you have to think that, for example, here I have a bit more A than B. So these peaks here, over time, will have a tendency to grow. This one also will have a tendency to grow. This one also. This one here, like that. So everywhere I have more A, I start to grow. But as I have more A, I'm going to produce more B as well. So basically here, I will start to build up a concentration of B molecules, here as well, here as well, here as well, and here as well. And over time, basically, this peak is going to rise, maybe this one as well. And since they're going to build up some B's here that maybe will sum with this one, they are going here to kill these peaks. So this one will disappear here. This one also maybe will disappear because of the B's that diffuse faster and are built on a longer wavelength. And here, this peak here will emerge and will go along the kind of long range of the B molecule here. And at the end, only the peaks will emerge at a very specific wavelength here. And the final pattern that you get is stripes in space here that are set, but actually this self-activation of A and the lateral inhibition due to the B molecule that diffuse away. So this is called actually diffusion-driven instabilities. And actually, I have a slide, I think, yeah, this is what I showed here. And that happens if you look at the mathematics of it. So you can write the whole system of equation. So here it's with U and V, but the idea is that you can write what is called a diffusion reaction equation. So diffusion reaction equation is quite simple, and you write the equation for the contraction of A particle, here over dt. You have a diffusion term, which is dA in 1D, dA squared over dx squared. So this is just to explain the diffusion. And plus, you have a function of the reaction terms. So this is all about the terms of reaction that involve A's and B. So this is where you will plug in this autocatalytic loop that will make it nonlinear in the sense that the more A you produce. This local production of B also. So this will be embedded in those terms here. And you have the same equation for the molecules B. Here. dB over x squared plus another function. So I don't write explicitly these terms here. You can find them on Wikipedia, on any textbooks. And they are very well-known schemes. There are a couple of them, actually, where you have different kind of non-linearities in the system that produce these diffusion-driven instabilities. So one of the requirements to have them is to have these conditions that are written here. So it's mainly related to the derivative of this function with respect to the A or B, U and V in this case. And you have these conditions that are actually pretty large. They are mainly related to the Jacobian of the system, of the reactive system. And the idea which is behind is that actually the chemical reaction, so if you remove the diffusion term here, you take only the reactive terms, it's going to be stable. It's going to lead to a stationary state. And the idea is that when you will add the diffusion, that will destabilize this stable reaction terms here. So to give you some idea, one way to understand how this pattern emerges here is actually when you look at steady states, what's the effect of this local self-activation and let's say global inhibition, is actually you can summarize that in a kernel, which will look like that you are going to apply on your concentration. So something that will have a Gaussian in the middle to say that you have this local activation here, and then you have this lateral inhibition, which you can have these lobes on the side that are negative here, and that shows that you have this long-range inhibition on the side. And that's going to give you the length scale of your pattern at the end, the size of that thing. So actually, this is actually a function that is used a lot by people in image processing, because this is called also like a difference of Gaussian, like a positive Gaussian minus a negative Gaussian. And this is used to do edge detection. So if you just apply that function on an image, you will enhance the contrast. You will enhance the border of the object here. And actually, this is exactly the same thing that we have in eyes. And this is an example I show you here. And actually, the image that you send to your brain, they are based on this on this lateral inhibition. So if you take the neurons on the retina, the first layer of neurons, you have a couple of neurons that are just beneath that actually do this local amplification and lateral inhibition. So it's like neurons that are not in your brain. They're really just beneath the retina, and they do what is called the pre-processing. They process the image before it is sent to the brain. And this is something you can do. You cannot do anything about it. Okay, you're built like that just to see better the contrast. And there are plenty of optical illusions that are based on that. And just for a little fun, I show you a couple of them. So this is one I like. I don't know if it works. So everything depends on the length scale. You know, if I am too close, it doesn't work, because actually in your brain, in your eyes, this length scale here is fixed. Okay, it's the distance between the neurons. So you have to get a good distance, but I think for the one in the back or in the middle, it will be good. You should see that each time you try to focus on a spot, the other spot on the side are getting dark. And this is due to this lateral inhibition. And you can fight with your brain, you cannot do anything about it, you know. It's like pre-processing. The one I like the most, and I will be done with this optical illusion, is this one here. Actually, the color of the A and the B are the same. So, do you trust me? Raise your hand if you don't trust me. Okay, I will do a demonstration. Is it still the case? Huh? Yeah, it is. And here, yeah, I'm still lying now. I'm still lying or not? No. Okay, you think I'm cheating, huh? But no, I'm not cheating. Huh? Seriously, I can do a flickering, but like here and here. Okay. So here, this one is really good because I'm cheating a bit. It's not only due to this lateral inhibition, it's also there's a second layer of brain interpretation about the context that enhances this effect. But at the end, it's also based on the same idea of this, the first cue that the brain is using is this lateral inhibition here, that enhances contrast. So just to come back to this Turing instabilities, actually, so if you take the whole mathematical systems here, we know that there are several classes of solutions for these systems. And here they are represented. You could have something which is uniform, stationary, something that will oscillate in homogeneously in space, but in time. you have some stationary wave with a very, very short wavelength, some oscillatory behavior of those stripes here, the waves that propagate through the medium, and the stationary one. So the stationary one is the one I discussed here, actually, this case 6 here. But you can think about many, there are other solutions. And here I show you in space some of these solutions. So you have this kind of spirals that can form. This is more related to the kind of Belousov-Zhabotinsky patterns we saw. Finger-like structures, fingertips-like, spotted pattern, etc. And one of the examples in biology that the people use a lot is this kind of pattern you see on the skins of animals here, where you can see actually beautifully the transition from one pattern to another one, depending on the growth of the animal, actually. At which time we started, actually? I forget. Let me look at the, sorry. Yeah, I should be done at, okay, I should speed up. So I will go fast. One example I like, I just give you the reference, you can look at it. I'll explain you in a second what it is, but I give you like some example of Turing patterns in biology now. One example I like a lot is this mollusk, where you go on the shell and they always have these nice stripes that you see. And actually how this comes from, there is actually an epithelium that is linked to a couple of secretory cells. The secretory cells are the ones that secrete the pigment that make them dark. And actually this pigment, these cells that deposit the pigment are connected to a layer of neurons that actually have encoded in their own behavior, in time, exactly this idea of this lateral inhibition and local activation. So exactly the same idea of this Turing kernel I was discussing before. So this is embedded in these neurons here. And actually what happens is that during when the mollusk is growing, you have this epithelium that is constructed over time that will then wrap up here. And then the cells deposit when the neurons here fire. So basically what you see on the outer shell of the mollusk is the thinking of the mollusk when it was growing. So you have the whole history of the thought of the mollusk that is imprinted on the shell. And actually, there is a very nice mathematical model that shows how the shape of the mollusk are related to these Turing instabilities, or HOP, so the temporal and spatial instabilities. The idea is a bit the same. There are some details that are a bit different. But you see that you can really reproduce, given the shape of those mollusks and the way they were growing, the whole pattern that you can see in nature. You know, these beautiful patterns like that. Or even like this is a mixture of a Turing and HOP bifurcation. Here it's just for Turing bifurcation, and this is for HOP bifurcation. So this is just to show you a very nice example. And then you have even more complex one with with wavy pattern here. Okay, so just one last example of one of these stripes here. And this is just to show you something about Turing patterns. It's actually, it's a good property for them, but a bad property in another case. Actually, what you see is that they are actually, the patterns itself, the scaling between, so the distance between each stripe is really something, as you can guess from here, that will depend on the dynamic parameter. So the ratio between the diffusion coefficient, the rate of the chemical reactions, but that doesn't depend on the size of the object. You know? So if you have an object that is growing over time, basically you will have at some point some doubling of the frequencies of this pattern, just to keep the length scale of the pattern being the same. So somehow the pattern is independent of the size, which can be good, but which is not good when you want, for example, as I say, to have a unique polarity axis that is going to scale with size. Okay, if you make a polarity Q like that with a Turing pattern, you know, if you get to a larger cell, you are going to get something like that, you know, which is not something you like and you want to. So this is one of the problems with Turing patterns. So this is like kind of the standard example that the people use in the community to show Turing patterns is mostly on, you know, animals' patterns on their skin. Another example that is good is actually the E. coli division. So I go very fast again here, but this is just to give you an idea. Where if you don't know it, actually the bacteria select their midplane by having an oscillation from pole to pole of a protein, which is called min C here. And this oscillation from pole to pole, when you look in average at the distribution of this protein, actually what you end up with, so if this is the size of the bacteria, you get two gradients here like that. You know, it goes from left to right, but if you integrate over time, and then you can find the midpoint very easily here. And to be fast, actually, this process relies on a mechanistic model where it's exactly the same as this kind of Turing-like idea, where you have a local self-activation and a global inhibition that is in place. And actually, if you take a minimal set of two proteins plus ATP and a membrane here, what happened, and you just look at what happens for this two protein, and you see this beautiful spatiotemporal pattern that emerges here, just thanks to that. So now you have to think, you can put a bacteria inside here, and then you will have a wave, a chemical wave that will reflect from one side to the other one here. Actually, it might be much more complicated than that, and there's still some debate about the system, but this is just to give you an idea about that. So there are some issues with the Turing-like patterns. They look beautiful, but actually, as I say, it's mostly for animal rope that we use them, and the issues that we have with the Turing pattern. So first of all, you need very specific nonlinear terms. So this is not something I explicitly said here, but actually these terms here are important. You need to have very specific combinations. So then you need to justify why you have those nonlinearities in the system, and this is not actually it's not a trivial task here. Something also is that they are very sensitive to parameters. Actually, so if you change slightly the value of the diffusion coefficient, dA or dB, or the reaction rate, actually you change completely the pattern, the length scale. So they're not robust at all to change of parameters in a region. So this kind of robustness that you want to assess is not easy to achieve with them. So also they tend to freeze, which means that once they establish, you cannot reorient them. You cannot put them in a new direction, for example. So in terms of plasticity, they're pretty bad. They cannot be. They don't show the plasticity you require for this kind of system here. Also, the patterns can be selected by the boundary conditions. So which means that to know if you are going to have vertical stripe of horizontal one, for example, because you don't know whether they're going to be in that sense or this one, actually then the geometry of the system count is going to select which one. And also on the growth, it can play a role as we saw before. So this is, let's say, about the details I was speaking about, the kind of all the robustness to the details in the system are not achieved neither here. So they're pretty good to have this spontaneous symmetry breaking, because as I saw, like a little fluctuation is enough. So they have a huge gain if you couple them to an input signal. So this is good. But also, like maybe a last point in which they are bad is that they don't have a null background, a null flat background level. So which means that, and not only that much here, but actually you want to have a system actually that is able also to be flat, to not be polarized all the time, because any kind of little fluctuation could kicks in. So in general, to have a robust system also, you need to have a state of the system that can be flat and that can be triggered by output signal. And you cannot do that with a Turing pattern, because it's always so spontaneous that it will always pattern by itself, not taking care of the rest of the system. So if you think about migrating cell, for example, it's not something that will be able to follow a cues from the outside here. So I will go now to another model, which is a bit based on the Turing-like, but more simple, which is, maybe I should actually do it here. It will be easier. which is the limited component model. So the limited component model, actually, the idea is to reuse this idea of local activation and global inhibition, but in a much more simpler way than the Turing idea. And the idea is really simple. The whole idea is to take a molecule that will still have some self-activation, but that is going to be limited. And that's it. And you're fine. So how that works, actually, so I have one experiment I like that shows how it works. Very simple one. Is actually, so it's an experiment where you have, an arena of 10, 20 centimeters, where there are some dead corpses of ants, okay, that are spread all the way around here. And there's a little hole in the middle here that is connected to the nest of the ants. And so the ants get in and they walk around. And what they do, ants actually they are a bit stupid, but also a bit intelligent collectively. They take, if they see a dead ant, they pick the corpse, they walk around, and then randomly they just drop the corpse a bit away. And the only thing is that, you know, they have a tendency to drop the corpse when there are other corpses nearby. And just that effect actually is enough to pattern and make scene theory. So how that works actually is the idea is that as the cells are going to move around, they're going to pack the corpse because they release the corpse, the dead corpse, more often. So this is kind of this self-feedback. You know, the more corpse you have locally, the more corpse you will put locally. And also, because actually this number of corpse here is fixed, okay, there is like 2,000, for example, to start with, then it's going to act as a limiting component. So this is the idea that you have a pool that is limited. It's not an infinite pool. And because of that, you are going to basically pack all the corpse into piles. And these piles, actually, you can do a model. They will have a very specific length scale, like an inter-pile distance that will be set by the dynamics of the ants here. And these are very lively patches. Actually, if you make a movie, you will see that these patches, they move around. One is going to disappear, another one is going to form here, maybe. So this is very highly dynamic structures here that are always constantly moving here. And this is pretty simple kinetics here. So again, with just this idea of a limited pool. So to give you some idea of where in biology we found this kind of model for cell polarity, the best studied example is the budding yeast, Polarity, where actually the yeast is going to make polar cues because of two processes. First, because it needs to divide to make a new to bud, to make a new yeast cell, and also when it does mating, when it needs to schmoo and find a partner for sexual reproduction. And actually, this first event of symmetry breaking that will define a patch somewhere on the side of the cell depends on actually an important player which is called CDC42. It's a GTPases. I will come back to GTPases a bit later as well. And the idea is that this patch of CDC42 is really the landmark of this polarity site on the yeast here. And actually, it was demonstrated over the year that one of the best models that recapitulates the data is actually the following. So you have to consider that you have the membrane, the plasma membrane here. And the cytosol. And you have a molecule that is going to be CDC42, actually. I can call it A, that will shuttle from the cytoplasm to the membrane. So you have the A membrane bound, A cytosolic here. Just shuttle in and out of the membrane. And then the idea is that the more you the species that is the A on the membrane is going to have a feedback on the rate at which A is brought to the plasma membrane here. So it's exactly this idea of the limited component I was speaking about, except that here you have also this idea of two compartments, the cytosol and the membrane. And this is really important because actually being on the membrane is the way you are going to have differences in diffusion coefficient. Somehow you immobilize the species on the membrane. And this is very important in any kind of model or physical mechanism by which you want to make polarity, is you want to immobilize, to have a very large differences in diffusion coefficient from the two species here. For the ends, it was easy. A dead end is actually not moving at all, so that was perfectly, that was direct. But in that case here, shuttling from the cytosol membrane is really a way to have something that diffuses fast in the cytosol and that will basically have a long range effect. And the membrane to be diffused slowly on the membrane is a way to really make patches locally somewhere. And so just with that, so this is the model that was proposed a couple of years before that you can see here. You go to the membrane, you diffuse, but slowly. Then you get out of the membrane, and there is this feedback on the rate at which you localize. Actually, depending on the amount of protein that you have here, this is like an example where you see a chemiograph over time. This is the contour of the cell, the whole contour here. And you see that you can start to form random patches that will be unique along the cell periphery for a given number of molecules that is in the low range. You see that plot here on the top. This is the red curve that you see here. You just form one patch. And this is due to the fact that actually, to make this patch here, you had to deplete almost all the molecules that were in the system. And of course, if you increase the number of molecules, then you reach a point where actually you don't get a unique patch, you get plenty of patch. And then this kind of polarity, which is only valid in a given range of the parameters here. But one of the beautiful predictions of that model was that actually if you take, so this is your wild type cell that will have one polar cap, then that will reduce to one bond. And if you have n molecules here, if you take a cell that now you engineer to have twice more of that critical component here, you should have more than one, let's say 2 or 3. So something that will be aberrant because it will start to make free buds. And this is the case, actually. If you do the experiments and you engineer such yeast cell, you do get several polar caps here.
So Mathieu,
yeah?
So in these examples that you described, the dynamics, well, the components are conserved. You have a finite amount of components. You're not producing some...
No. If you're in a regime where you produce or you maintain at the level, at a given level by synthesis and degradation, a given amount of protein, the problem is that here it's like a chemostat. Okay, you maintain that number, whatever happens behind, and that's not good. But
maybe you would have other regimes where, in fact, like driven regimes where you have this patterning but driven by production of some component.
Yeah. And that's in a different regime, yeah. It depends on the time scale. Somehow the polarity I'm speaking about here, there are processes that are quite fast, actually. They happen on a time scale of a couple of minutes. And when you look at the time at which normally you have this homeostasis between synthesis and degradation, is over hours. You need like kind of synthesis of gene synthesis and protein degradation. In most of the models here, they assume a constant amount of, conserved amount of...
in general, those patterning or polarization, they sometimes occur driven by some production of some gene or something.
Yeah, of course. So this is...
So you have the different possible dynamics.
Yeah, you still have the possible dynamics. Yeah, I am not going to discuss all the models, of course.
No, just to make perspective.
Yeah. So still, actually, these models also have some issues. So that was for the Turing here. The issue that you may have for this one, I'm not going to list all of them, but for example, it's not really robust to the concentration. So if you change the concentration, as I say, you change the amount here, you're going to change completely the number of, you don't get something that is unique at the end. So this is not robust to that. Also, they tend to drift. Sorry. So whereas like a Turing pattern tends to freeze, it stays where it is and that's it. This one tends to drift because, you know, if you look over time here, actually, since it's a dynamic state that is maintained as well, it can drift and move around. So if you want to keep a patch somewhere and that don't move, it's not very efficient. So actually then there is like, but the time is running, so I will go fast. Other models. So just to give you one example here, there is plenty of other ones that could be driven by gene expression, for example, like dynamics of gene expression. But here is one that is based on contractility. So it's an idea a bit like Turing, where you have an instability that will drive the whole system into a new state. But here, instead of diffusion-driven, it's like contractility-driven. I don't get into the detail, but this is a bit the same idea, where you are going to have something that will be destabilized by contractility here. You have another model that is nice, and that is called the wave pinning. I just mentioned it here. Wave pinning. So the wave pinning model is actually based on a bistable system, like we had this morning, like Mark introduced. It's a bistable system that has two states. You can do that with, again, this idea of a non-linear production slope with like linear decay with two stable states and one unstable state. And if you add that and you put diffusion on top of that, and plus a limited component, so something that will be consumed, actually you can have a wave that will propagate, that goes from the low state to the high state, but that will be stopped by the fact that you consumed everything. And then that it will stay in position, and that will do this kind of what is called a wave pinning. Yeah. That was a bit popular at some point here. So more than going a bit further here, I will just get the last couple of minutes to just say actually what is the tendency now. I think one of the tendencies is to say that actually all of these models have issues. Actually, there is no unique model that can actually fulfill all of these processes here. And because of that, people stop thinking about a unique model that will recapitulate everything. That is not possible, let's say. But rather, they think about modules, you know. They think about some models that will do the symmetry breaking, for example. So this idea of spontaneous, they just say, OK, I need something that will just break symmetry. Some of them make models that does only the scaling. They will only scale. Other one, they do this sensitivity. So, for example, for the plasticity, they do adaptation to different environmental cues and sensitivity to incoming signals. And the idea is then build a mixture of these different models here. And in the last couple of minutes, I will present you the model that actually we developed. That is a bit based, so still something that is really important is actually this cytosol and plasma membrane is something that will always come back, is really to have an exchange between cytoplasma membrane is a way to get these very large differences in diffusion coefficient that is in mobilities. That is something you need if you want to make a pattern at the end. And so one of the questions that in terms of the model, that what we ask to the model, we can call it like the, I don't know how I was calling it. Yeah, let's say the coordination. I say about symmetry breaking, scaling, all of that. So what I mean by coordination is that when you have a cell that, let's say, have several patches of activity, and you want that all these patches are going to talk to each other, such that they are going to resolve into just one patches that will be, let's say, for example, if it's a cell that migrates in the front here. So how you can couple these different patches of activity, so this is the kind of coordination. So again, there was like a discussion about different models. And one experiment to show that, how that happens, so that I found fun, is actually a cell that is growing after a heat shock that make actually a small body and a very large protrusion very far apart from the cell body. And actually, you only have one protrusion that is active and the rest is inactive. And actually, when you cut with the laser this protrusion, you see that the body that was quiescent starts to make a new protrusion. So somehow there was some information about in the body here, coming from here, telling that I have one active front and not everywhere. So this is really this idea of coordination. One thing was telling to all other places of the cell still be quiescent. And there is like a big discussion, so how this is achieved. And what we propose for that is based on the raw GDPases. I will go fast on that. Yeah, just look at this. Don't look at everything, it's very complicated. Just look at this image here. So the raw GDPases are actually a small molecule that are either in the GDP form or in the GTP form. So they are activated by some proteins, inactivated by other ones. They shuttle. And there are three canonical of them, Rho A, RAC1, and CD32. And actually, RAC1 and CD32 are known to be active at the front, so really like that patch here. And Rho A is known to be active in the back here. So since they have this pattern, and we know that there are some relation between both, the general idea was to say that, oh, there is a crosstalk between both, and they exclude each other to make this pattern here. And actually, I go fast, but I show you what we did, which is based on this experiment here. What we did was to use optogenetics to activate Rho A locally. So actually, we took a cell that was not polarized at the beginning, and we actually impose a patch of activity somewhere. And actually, what we saw when we did that, oh, I'm sorry, I'll show you the movie. What we saw is that actually, when you activate, the cell actually has some contraction on one side, but at the same time, if you look at the other side, the cell extends and protrudes. So you have really like a movement of the cell in the other direction, because actually there is a kind of automatic response of the cell. As soon as it retracts, it protrudes on the other side. So which is really like how there is a coordination between this back and the front, and by which mechanism is it achieved. This is what we were wondering, and this is highly reproducible. You can see for many cells that you always have this back that retracts and the front concomitantly. And just to make a long story short, because I'm done, actually the model that we had is actually based on this very simple scheme here, but even more simpler than that. And I will explain you in a second the model. We don't have this feedback here. What we assume is that there is an exchange between the membrane and the cytosol here of two components. I will explain to you like actin and myosin. And the idea is that we assume that we have just two boxes, let's say, what we call the back of the cell, where we do the activation by hand, so we force that, and the front. And so at the beginning, you have an equilibrium of your species between the membrane pool and the cytoplasmic one. And actually, what you do when you do optogenetic activation, so when you overactivate these rhodopsiases at some place, you are going to imbalance the kinetics. And so you are going to make a big relocalization of the cytoplasmic component here on the membrane, which means that actually the balance between the cytosol and the membrane will be changed here. And that will affect, since this one shares the same pool, actually it's the same cytoplasm, they are going to affect the balance here. So this arrow is going to be decayed, is going to get lower, and this one will be reinforced, which means that you are going to deplete the level of the membrane-bound form away from the back. And this is something you can simply express. I will just show you that. Just to show you how the biological relevance of that is actually, this happens also when two cells collide. This is called the contact inhibition of locomotion. The two cells collide and they tend to repel each other. And this is based to actually to local activation of Rho A at the site of contact, and at the same, exactly the same time, a formation of a protrusion on the other side here. And I show you very directly that will be the N here, the idea. So this is the model. So you have these two boxes, I say, and this is for actin and myosin. So actin actually is this cytoskeleton that is within the cell, and myosin is the one that contracts. It's a molecular motor here. And both actin and myosin are either cytoplasmic or membrane-bound. And actually, when you are going to activate in the back, you are going to deplete on the other side. And you can compute this effect of homeostasis between the front and the back, okay, by one number, which is actually only dependent on the rate, the equilibrium at the beginning between the rate at which you go to the membrane and the rate at which you dissociate to the membrane. And this is quite simple. If you just take this even more simple equilibrium between the membrane and the cytosol, you have the membrane-bound form of A here, over dt. Here it's not even spatial. I just take one box here. It's going to be the K on, so if this is the K hot, and the K D for the deactivation or decay from the membrane. K0 times IC minus KD AM. If you have a conservation law that tells you that the total amount is the sum of the cytoplasmic plus the membrane bound, you replace it inside, and then you just get this K0 A0 minus AM minus KD M, and a steady state. If you look at steady state, you just get that AM, I divide by A0, so this is the fraction that is membrane bound, is just simply the K0 over K0 plus KD. This is as stupid by that. So I can divide everything by KD. And you see that this is just 1 over 1 plus the dissociation rate, which is just dissociation rate, the KD over K0. So this is just to tell you exactly just one number, the ratio between the two rates, the on and off rate from the membrane, tells you how much you have on the membrane. And here is exactly the same. If you look, you can do the calculation, and you say how much you deplete in the back versus how much you put in the back, and how much compared to how much you deplete in the front. You just look at that from the very basic chemical reactions, exactly the same one as I wrote here. Okay. You find that this number, which tells you the kind of homeostasis, that is a number between 0 and 1, is just going to be a function of these rates here. And actually, the more you are going to be on the membrane, okay, the more you are going to be on the membrane, the more you are going to have this effect. And 1 is maximal, so when everything is the membrane, you have the maximum balance between the two compartments here. And that's the whole idea of this model. So, and I think what is nice here is that it actually depends on almost nothing. Okay, it doesn't depend on the concentration. You can double the amount of concentration in the cytosol here. If you assume that you have enough space on the membrane to account for that, okay, if you double the number here, you are going to have twice more on the membrane. But that's fine. The ratio is going to be the same. And this effect, this coordination between the front and the back is going to stay the same. And so that's what it means. It don't depend on any kind of number, just this rates, the balance between the membrane bound versus the cytosolic form here. And to show you how it works actually in terms of how the cell moves, why it moves forward, actually what it does, you can think about the cell having a shape where it's a balance between contractility, which is dependent on rho A, that tends to contract the cell, and protrusion, that depends on RAC and CD42. And it's the balance of both that give rise to the shell shape. And if you block contractility by a drug here, actually if you block contractility, if you do the experiment, you see the cell that actually extend from everywhere. And actually when we just activate locally actin and myosin, we relocalize actin and myosin through this effect that I say. So basically you increase the amount of myosin and actin locally, but you also decrease it on the other side because of this mass balance. And by doing that, actually, you are going to contract the cell here and let it protrude on the other side, just because you reveal this protrusive activity here. And that's my last slide, I promise, is more on the more general modeling aspect, actually. The idea of this model that has this mass balance connection between two different places in the cell. You can tune it because, you know, depending on this, this number here, which tell you what is the fraction bound on the membrane, either when you're in a case where you have plenty of cytosolic and not that much bound, and in that case, you will have plenty of spot of activity that will be able to coexist. And if you want to all of them resolve in one spot, one way to coordinate them, as I say, is actually to unbalance toward the bound forms, where you put everything on the membrane, and that will be a way where you can end up with a unique polarity axis here. So you can really coordinate independently of the cell size and amount of concentration or whatever. It's a very simple way to do it. But actually, the payoff that you have to do is that because you put a lot of membrane, it means that you have a very low or slow turnover. You don't have that much molecules that are unbinding. And because of that, actually, the overall dynamics can get longer. So, and what will tell you how long it takes, for example, if you have a new signal to reorient, will depend on this turnover, which is actually given by this KD here. So you have a balance at the end between plasticity, how fast you want to adapt, and coordination, how strong you want your coordination between different positions in the cell to talk to each other. And I will stop here, and thank you for your attention for this, I know, hard end of the day.